Now, at the end of the last mini lecture, we had talked about the Gibbs free energy of the individual reactions and how we can modify that based upon concentration. Because this is effectively like an equilibrium constant, products raised to their power divided by the reactants raised to their power, we can plug this into our reaction quotient. And what that means is altering the concentration can make a reaction that is non-spontaneous spontaneous. Now, where this is really critical deals with glycolysis. Glycolysis is an incredibly lovely synthetic pathway that converts a glucose molecule into a number of pyruvate molecules. The net reaction, it has in the process, it releases energy to couple the reaction to form ATP. Now, we talked about coupling reactions. When you have two reactions, one that is non-spontaneous and one that is spontaneous, sometimes you can use the spontaneous one to drive the non-spontaneous one. This is going to be very critical when we talk about electrochemistry. We have complementary reactions where one is non-spontaneous under normal conditions and then spontaneous under other conditions. And those two reactions couple together and are driven towards complete reaction. 10-step pathway, in each of these steps has its inherent Gibbs free energy. And so if we actually just take a look at another graphic, we can see this Gibbs free energy documented where the blue would represent the individual Gibbs free energy under standard states. We see that a number of the steps are highly energetic under standard state conditions, these blue labels. But we also see that a number of them are highly non-spontaneous. And that means that the reactants are very much favored, and so the reactions to progress. So we can talk about the individual steps that lead up to this process in this fashion. But that's only part of the picture. If we look at, though, what happens actually under physiological conditions, under molarities of starting materials, what we see is that a very different picture. We see that almost every single one of the reactions, which was non-spontaneous, is now spontaneous, and significantly spontaneous. Or they're run at almost equilibrium conditions. So reactions that were non-spontaneous, are effectively non-spontaneous, are now all spontaneous or equilibriums. What this means in terms of glycolysis is that many of the steps in glycolysis can be run backwards as well. So that means that the reactions take place near equilibrium, allowing them to also be driven backwards to regenerate the products. This allows the biochemical pathways to recycle the materials that are in excess, driving them into different chemical. It's not just what the Gibbs free energy says, it's also what the Gibbs free energy of the reaction is under non-standard state conditions. And so we have to take that into account.